All right, everyone, hello. It's great to see you all. I hope you all are doing great. Welcome to FAU Harbor Branch Oceanographic Institute and the John and Barbara Ferreira Ocean Science Lecture Series. My name is Dr. Gabby McHenry. I'm the Director of Education and Outreach here, as well as your host. As many of you know, this program has been part of the Institute's outreach for nearly 30 years, and it allows us to share some of the amazing work that we do here and around the world. So thanks so much for being here. We hope you enjoy and even learn something new today. Before we get started, I want to take a moment to recognize everyone who supports FAU Harbor Branch. I'd especially like to recognize John and Barbara Ferreira, as well as those who donate to the Ocean Science Lecture Series and make evenings like this possible. Community support impacts so much of what happens here, from the research taking place behind the scenes to the education and outreach programs that we share with our community. So as you listen throughout the season, you might find a topic or project that sparks your interest, and when that happens, there are many meaningful ways that you can get involved, from sharing what you've learned with others to volunteering your time or making a gift in a way that's right for you. And there's some options behind me on the screen, and I'd be happy to chat with you if you're interested. I also want to take a moment to thank all of the staff here who support the series and help, as well as our speakers. Of course, it wouldn't be possible without them. And to get started here, I've got a couple of general announcements. First and foremost, please take a moment to silence your cell phones. We are in the final month of our 2026 season, so as a reminder, lectures take place each week through March 25th. Your registration covers access to all of them, so no need to sign up again, but please do encourage your family and friends to sign up and join us as well. Each presentation is also available virtually, so hello and welcome to everyone tuning in on our live stream. And the presentations are recorded, so if you miss one or you really want to see one again, you can do so on our website. And for everyone here in person, as you also know, each week we offer a reception in our gallery with our speaker and some cookies. So please do join us for that and strike up a conversation with some of our staff members to learn more about all of the great work that we're doing here. And while you're out there, be sure to visit Jen at our pop-up gift shop and take some flyers to have more information about our programs. And finally, be sure to mark your calendar for next week's lecture on March 18th, which is titled Cell Imaging to Reveal the Hidden World of Plankton by Stephanie Schreiber and Olivia Rucci, who are two graduate students working on their Ph.Ds here at FAU Harbor Branch. We're excited for them to share their research with you, so please be sure to join us for that. And that brings us to today's presentation by Dr. Wayne Slade, which is titled Ocean Optics, Shedding Light on the Ocean Around Us. Dr. Slade has been an associate research professor here at FAU Harbor Branch since 2023. Before joining FAU, Dr. Slade spent a significant time in the private sector with Sequoia Scientific, Inc., where he led instrumentation research and development for ocean optics and other environmental and industrial applications. He received his Ph.D. in oceanography from the University of Maine in 2011, focused on ocean optics, and before that received his bachelor's and master's in electrical engineering, working on neural networks and machine learning applications to problems like ocean color remote sensing. Dr. Slade's work bridges engineering and oceanography, working to better understand how optical tools can be used to infer biogeochemical and physical processes, as well as designing and deploying optical sensors and developing new methods to measure optical properties in situ using a variety of platforms. At FAU Harbor Branch, he's establishing a new lab focused on optical particle sensing relevant to inland, coastal, and open ocean processes, as well as applying these tools to understand specific events like freshwater releases, harmful algal blooms, and storms that have a significant impact to coastal Florida and beyond. And he's going to share some of that work with us today, so please join me in welcoming Dr. Slade.
Thank you, guys. Thank you, everybody, for being here. Thank you, Gabby, and all the outreach people for making this lecture series possible. Also, the donors that make this possible as well. Thank you for the people online as well for tuning in. in this virtual world. Like Gabby said, my background is both oceanography and engineering. And in general, I study ocean optics and how you can use optical techniques to study especially particles in the environment. Um, yeah, and like she said, my background's also engineering, so I tend to be really interested in technology development and that sort of problem. As you might have guessed from the title, today I'm going to focus on ocean optics and mostly how we use ocean optics to look at the ocean from above using remote sensing. I'm kind of hoping this is like a 101 level or maybe sub 101 level look at ocean optics with no math. I'll try to avoid saddling you with any, without too much complexity and try to show some pretty pictures of ocean color along the way. All right. And we're off. Oh, yeah, and it's kind of a little bit of a last minute change. We use ocean optics for a lot more than just looking at the open ocean. We use it for coasts, lakes, lagoons, rivers, and so on. So, but it's kind of makes the title a little bit long. I also just wanted to throw out this last month of the Ocean Science Lecture Series is kind of a little bit thematic to me. I'm talking about optics today and light, which is an electromagnetic wave. And next week, our graduate student lecturers are both using optical tools to look at plankton and phytoplankton, and they use light, so that's wave phenomena. And then finally, Dr. Matt Ajamian wraps up the lecture series with a talk about underwater sound and acoustics, which is yet another wave. So just thought that was kind of thematic. All right, so Dr. Tim Moore here at Harbor Branch gave, I think back in 2023, a really good ocean science lecture series talk about a brief history of NASA's Ocean Color Satellite program. And he started out by connecting it all to these first photographs that NASA Apollo astronauts took of the Earth in the 1960s. For many people, this was the first time that Earth was seen as a whole system. This image, very simple image we think now, but it revealed oceans, clouds, and continents and weather patterns all interacting on a single planet. With scientists and engineers of the time, it really served to galvanize the idea that we could use space and remote sensing and satellites for Earth observation. We realized that we could view the Earth from above and get really powerful insights in how the planet works. From space, the planet appears mostly blue, so you may have heard this first image called the blue marble. And that, again, blue, it just really highlights how much of the Earth is covered by the ocean and emphasize the global importance of oceans to us as humans. I think Dr. Moore uses the famous Earthrise photo, but I really like this one from 1972, which is one of the last ones taken, if not the last picture of Earth taken during the Apollo program in 1972. And I like it. I like this unprocessed frame, just how it was taken sort of off-center on Kodak Ektachrome film before it was cleaned up and enhanced and used for public uses. Contrast that with the image on the right. This is an updated NASA blue marble. This is actually where they use modern remote sensing instruments that have been developed in the 5+ decades since the original blue marble, and actually stitch together views of the Earth. And this is one day. We get this sort of coverage every day, multiple times by different sensors that measure different things. So it's really a phenomenal capability that we have as scientists and engineers now. So what is ocean optics? Let's just start there. Start with optics. Optics is the study of light, how it's generated, how it propagates in media, how it interacts with different materials. Light itself is electromagnetic radiation, like I mentioned earlier, and specifically it's radiation that's visible to the human eye. Just a side note that we often include ultraviolet and near infrared when we're talking about optical sensing. Now, ocean optics specifically is the study of how light interacts with water and the materials within it. Like I kind of indicated on the title slide, although we often focus on the open ocean, the field really broadly includes rivers, lakes, inland waters, nearshore coastal systems, but we still use that ocean optics term. To add a little additional context to the field in general, we need this knowledge of ocean optics and how light interacts with materials in the ocean so that we can understand what information can be extracted from the color of the water as it appears from satellite sensors in space. While this ocean color is a major theme of ocean optics, it's really a broader field that also includes development of sensors using optical methods to study more than just the color, polarization. We use these sorts of techniques to understand chemistry, particle properties within the ocean. We ultimately use this light to image things like plankton and bacteria and other larger organisms. So it's really a huge field. It also includes things like diver visibility, underwater visibility and detection, and even bioluminescence, which is looked at pretty significantly here at Harbor Branch. But today I'm going to focus on ocean color, which is necessarily my bread and butter day to day, but it's part of it, and I thought it would be interesting to put together some stuff. So going back to the idea of Earth observation, I'm not going to go into each one of these little dots, but this is the current global civil fleet of Earth observation satellites. This is just to give you a sense of how many satellites are up there since that initial NASA Blue Marble photo on, with I think a Zeiss lens and Ektachrome and Hasselblad. But there's so many different things that are measured, ranging from our gravitational field, different physical phenomena about land, ocean, atmosphere, fires, ice cover, all sorts of things. And these views are just, I think we kind of take them for granted for today, but it's only been 50 years that we've been developing this technology, and it's pretty astounding. Okay, zooming in on a little bit to look at what NASA actually has in orbit right now. These are the satellites. Again, not going to go into everything here, but just want to indicate that some of these specific products that we get in the ocean are relating to phytoplankton biomass, phytoplankton diversity, different particulate matter properties, particulate being sediments and things like that, dissolved organics, sea surface temperature, sea surface salinity, ocean currents, sea surface height, which sea level rise and all these things, and waves. So there's just a huge, pun intended, a huge swath of things that we measure with these satellite sensors. Some of these same sensors also measure things like cloud properties, vegetation on land, ice cover, fires, like I mentioned. So just a really wide range of things that we get from these sensors. So I mentioned ocean color, and some of you might be familiar with this if you've been to other talks here or read about it before, but basically this ocean color remote sensing relies on the idea that plants have chlorophyll. And that's the pigment inside leaves, like when we think of terrestrial plants that make leaves green. And that's because that pigment mostly absorbs blue and red light and allows green light to be reflected back to us, whether that's our eyes or a remote sensing instrument. That same chlorophyll pigment is in phytoplankton in the ocean. Phytoplankton are microscopic plant-like organisms that drift through the ocean. Just like plants, they use chlorophyll to capture sunlight and grow, converting it to energy and biomass. Basically, when there's lots of phytoplankton, the ocean goes from blue to green. And we know this sort of intuitively by looking at the ocean and when there's blooms versus not blooms. But satellites can detect very subtle differences in that color difference. So these satellites work like very sensitive cameras, and they measure specific wavelengths of light coming off the ocean surface, and they are able to pick up these very subtle differences. So one, for example, global satellite can scan the whole globe in just a day or two. And that gives scientists and policymakers a near real-time picture of ocean health, which would be just impossible if we only had research ships to go out and actually take physical measurements. So we've been collecting these data for a long time. So I think ocean color started around 1978, 79 by NASA in an early experimental proof of concept mission. But we still look at that data occasionally. And this marble here is just a record of that chlorophyll, both ocean and land biosphere measurement from 1997 through 2017. So the colors represent the amount of chlorophyll. So these purples that you see here in the ocean gyres are very low chlorophyll, and then you can see the seasonal effects of this one's really purple. That's the South Pacific gyre, really cool water, very dark or very blue. But you can see these patterns and basically the Earth breathing as phytoplankton grow and take in CO2, and it's just fascinating. You can stare at this as an oceanographer. You can stare at this globe for way too long. Okay, so I mentioned wavelengths and all these things. One of the keys, if you get anything out of today, one of the keys is that white light is actually, like white sunlight is actually a mixture of many different colors. And we've all seen Dark Side of the Moon or maybe science kits and actually seen a prism and put light through it and see that we can disperse that white light into different wavelengths. Basically, the prism here separates, each individual wavelength of light bends slightly at a slightly different angle through the prism, which causes the separation and creates this Roy G. Biv rainbow sort of spectrum. And this is key, that each one of these different colors, what we perceive as colors, are different wavelengths of light. So put that in your pocket. You'll see these the rainbows on figures in the future. This is the bigger picture of what electromagnetic radiation looks like, just to kind of give you an idea that visible light's actually a really small part of electromagnetic radiation. It includes other stuff like radio waves, X-rays, gamma waves. But when we talk about visible light, we're referring to this really tiny part of the spectrum that conveniently is in an atmospheric window so that light can get through. That's why we get sunlight. And the wavelength of these waves in this region are in the probably 400 to 700 nanometers is what we generally consider to be the range of visible light. And as context, a nanometer is 1/billionth of a meter. Think of a meter like a yard. So a totally useless comparison would be that our fingernails grow about 1 nanometer per second. So think about that. But maybe a DNA molecule is, I think, 2.5 nanometers wide. So that's a little bit better context than a fingernail. But again, we perceive these different wavelengths of light as different colors. That's really 90% of ocean color remote sensing. Okay, so when there's no obstacles in the path, light just travels in straight lines that extend in all directions from its source, kind of like an expanding sphere. Once light encounters an obstacle, different things can happen to it. And even outside of science, we hear a lot of these words like light being transmitted, or things, light or heat being absorbed or reflected or refracted or scattered. So just to somewhat formally define all these, transmitted light is just light that passes straight through an interface. So it's transmitted through a transparent surface in a sense. Absorption, however, is one that's really important for us. That's when photons from incoming light hit atoms and molecules in an object. That object could be, for example, a phytoplankton cell, and through complex physics basically causes things in that cell to vibrate. That vibrational energy is converted into heat and chemical energy, and that light is just gone. And that's, again, like, that's probably, I guess, wavelengths. Maybe let's call that 80% of ocean optics. 10% is the idea of absorption. Another 10% is scattering. Ocean color, there you go. When it hits a surface like the ocean, some of that light can get reflected. It can refract into the ocean. Those are sort of processes that we won't talk about too much today. Diffraction is when light hits either a narrow obstacle or a slit, and it expands past that obstruction. We won't really talk about that explicitly. But scattering is when light is redirected. So like if that light hits a water molecule, grain of silt, phytoplankton cell, some of that light can sort of bounce off it in any given direction. A lot of what I do is trying to figure out how that directionality and how that bounciness happens, and how we can design sensors to use it to our advantage. So, okay, we're going to focus first on absorption, and these are our big primary absorbers that we like to think about in ocean color. These are phytoplankton. They're single-celled plant-like organisms. They drift through sunlit surface waters of the ocean. They convert sunlight and carbon dioxide into energy, cellular biomass, and oxygen through photosynthesis. They're the base of the aquatic food web, and provide food for all sorts of sea creatures. The collage on the left here is just to give you a sense of all the different variety. We actually have several speakers that give excellent talks on phytoplankton because it's their background. But this is just to get a sense of the variety, and I think next week these images are actually from an instrument called the Imaging Flow Cytobot that we have one of these here at Harbor Branch, and I think one of the graduate students will be talking about it. So you saw it here first. So again, this is just to give you an idea of the sheer variety of things out there. The two on the right are two other species or groups of different phytoplankton that are important for ocean optics, ocean biogeochemical cycling and such. The one on the top is a type of phytoplankton called Prochlorococcus, and it's the most abundant cyanobacteria in the ocean. It's one of the major primary producers in the ocean. It's really small. Each one of those cells is like 0.6 micron or 600 nanometers in diameter. The lower image is a type of phytoplankton called a coccolithophore, and these are really interesting because these plates, these little, I mean, they look like plates, are made of calcium carbonate, and these coccolithophores both shed these plates, which creates very, very fine particles of highly reflective chalk in the water, so they're very visible optically. So they're also very important biogeochemically because that carbonate is, they're a very important part of the carbonate cycle in the ocean. So yeah, so this diversity matters a lot in ocean color, and it's one of the things we aim for in ocean color is to try to understand what kind of phytoplankton are out there. Both how much, that's our first order problem, but then can we figure out what kind it is? And, or at least know how those differences could confound our estimation of the total amount. These are some examples of algal cultures. These are from the Bigelow lab. They have a big culture collection. And you can see these cultures have visibly very different color. And that's because each one of these different cultures is a different species of phytoplankton. And each one of those different species has different groupings of pigments. And this plot is showing absorption. So absorption versus wavelength. So the wavelength of light from 400 to 700, so from purple and blue end of the rainbow to the red end of the rainbow. And we mentioned chlorophyll. Chlorophyll's right here. And basically all of these pigments, most of them at least, tend to absorb in the blue and absorb in the red, and that leaves light in the green. So typically we see plants and phytoplankton, most of them are greenish. There are some that have these additional pigments that absorb a lot of the green light, and so that leaves a lot of red light, or like sort of the lower end of the red light. So that's these ones that look, for example, pink. So these are spectral signatures that we can use. If we can try to understand what our phytoplankton absorption looks like, we might be able to tell what kind of phytoplankton is there. That's one of the big questions in optical oceanography and ocean color. Okay. So another thing in water that's really important is called dissolved organic matter, or colored dissolved organic matter. And you can think of that like tea or coffee. Basically, this substance absorbs very strongly in the blue and doesn't absorb very strongly in the red. And it's basically in the environment, it's essentially making tea from things like mangroves and vegetation and trees and soil. So it's all of that stuff in the tea bag of nature that makes things like the Indian River Lagoon and other coastal systems have a tea-like color at times. It's also largely responsible for the color of Lake Okeechobee, for example, if you've ever been down there. So pretty important stuff. Okay, so going on to scattering, why do we care about scattering? I mean, we'll talk about it in terms of ocean color, but just thinking about it conceptually, we see this diver, this diver that's having a rough go, and scattering is what's really decreasing the visibility there. We see some green color because there's a lot of phytoplankton and such, but all those particles that are in the water are scattering a lot of light back to us. So to give you another look at scattering, this is 2 beakers and a laser, and the first beaker has basically just clean water in it, and the second has some very small particles, but fairly dilute. And you can see that in the beaker with no particles, or very few particles, we basically can't see the laser. But when we add particles, we can see the laser. This is basically what happens in the ocean. We need scattering for the light to get back to our eye or to the remote sensing instrument. So that's why scattering is really, really important. It also helps us explain both of these phenomena. It helps us explain why the ocean's blue. When sunlight goes into the ocean, it interacts with the ocean water itself. So if you don't have phytoplankton and such, water itself absorbs very strongly in the yellow and red, and it scatters in the blue. So what's left over, what's reflected back to us after the water has absorbed the red, but then also scattered what's left back to us preferentially in the blue, the reflectance that we see, which is, you can think about as the ratio of backscattering to absorption, is blue, very blue. And this is actually, it's not my photo, but it's a photo from the Indian Ocean, and it's just got that really, really rich ocean blue, very, very clean water. If you go to the South Pacific Gyre, places like that, the ocean gyres, there's even less material, and it can even start to look purple. It's an absolutely incredible visual. Okay, so big picture of what's happening with light entering the ocean, just to give you a little bit of a overview. You know, we have what's called the euphotic zone, which is the sunlit surface of the ocean. And then as that light is being absorbed and scattered, it decreases as a function of depth. And we go through the twilight zone, where there's some light, but you know, you really can't, like, stuff can't photosynthesize, but there's a little bit of light. And then you go even deeper and you get to the aphotic zone, and there's just no light. We now know that there's a lot of organisms that create their own light for all sorts of different reasons, but we don't see that today in the satellite images. Another way of looking at that is in terms of the spectra of that light. Right here is the the spectrum of the incident sunlight from 400 to 700, and it's pretty flat, but it's a lot of blue light that's coming in. And when it goes into the open ocean, it's absorbed kind of as we showed before, and your light looks pretty light bluish. But then if you go to the coastal ocean where we have a lot of material we'll talk about a little bit, that light is being shifted to the yellow, towards the yellow, green and yellow, and a lot less deep because that light's being scattered and absorbed on its way through the water column. Okay, we won't dig into the intricacies of this figure, but this is sort of a summary of ocean color remote sensing, where we have sunlight going into the water, interacting with material, and some of it making back to a remote sensor. That could be us looking at the water, or it could be a satellite in space, or an aircraft, or some other sensor. The things that can happen to that light when it gets into the ocean, basically it can be absorbed or scattered by material. And the materials that we're typically concerned with are these dissolved organics, water molecules themselves, which scatter and absorb, and algal pigments, which absorb different colors of light, and then other suspended solids, non-algal particles, so they're detritus, so like dead phytoplankton, fecal pellets, silts, and sediments and such. So those are the things that are affecting our water. This is a true color image from one of our ocean color remote sensing instruments looking at a bay up in Quebec. And I just show this because in this one scene you can see a wide range of different optical environments in the ocean. Offshore here, you have something that's closer to our blue water, where there's not a lot of sediment, not a lot of dissolved organics, and the water looks blue. Out here, we get some mixed absorption and scattering by CDOM and non-algal particles. And then here in the river flow as it's coming out, there's actually a lot of CDOM, but then right here, you also have a huge amount of sediment that's been resuspended. And you can kind of think, just to fast forward here, you can kind of think this is a similar situation. It's kind of like adding milk to your coffee. Here is the dissolved organics. This is the confluence of two different rivers in Brazil. This is the Amazon, and this is the San Mateo. But it's a well-known river confluence, and you have this very tea-like water coming from this river mixing with the Amazon, except it doesn't mix initially just because of the physics. But as it does, it's interesting because the Amazon has all these scattering particles in it. It's kind of like when you add milk to your coffee or tea. Those are particles that take your coffee and tea from being that dark absorbing liquid to something that has lighter color. And we see this sort of thing in the Indian River Lagoon too, with sediment resuspension because it's so shallow, and lots of outflow of organics. Okay, here's another one just to kind of give you two different optical worlds side by side. On the left is an image from a recent cruise in the Sargasso Sea, so it's pretty blue water. This is a lot like that water that I showed you earlier, looking at why the ocean is blue. There's not much phytoplankton here. There's a lot of absorption in the red and a lot of scattering in the blue. So we see it as nice, beautiful blue water. And then there's something that's a little bit more, let's just call it coastal on the right. You could think of this like the Indian River Lagoon, or some sort of similar environment. So here's our situation in the blue. This is very similar to before, except now we have just a tinge of other material in the water, some phytoplankton, some CDOM and such, and a little bit of material scattering, but it still gives us largely that blue color. And if you contrast that with with this is the situation. This plot is basically just sort of stacking these different substances on top of each other. So here, that blue is, again, the water, but now we've got a ton more dissolved organics, that T, that's absorbing in the blue. And then we have a lot of scattering just across the whole spectrum. So that actually is making the water look brown, right? We've had all of our blue light taken away, and we're getting a lot of scattering across the rest of the spectrum. So if you think we've got this peak of the reflectance coming back to us is in that 600-ish nanometer range, and that's yellow. It's yellow and with a lot of light over here into the orange and red. So it looks brown to our eyes. So that is a summary of ocean optics right there, or ocean color at least. This is sort of thinking about that blue water as you don't add a huge amount of sediment, but you just start to add phytoplankton and chlorophyll. You see something similar where you go from very low concentrations of chlorophyll in the black and blue to high concentrations of chlorophyll in the orange and red. And as you go from this very blue water, as you add chlorophyll, you're now sort of shifting the peak of your reflectance over to the green, right? Where now we've got a peak of light here around 550, and we're solidly green. So that's just kind of a hand-wavy description of how we measure ocean color, how we can use ocean color to measure something like, to estimate something like chlorophyll. We can use both chlorophyll algorithms like that and other similar algorithms to look at all sorts of different processes in the ocean. We can like productivity, we can look at water movement, sediment loads, harmful algal blooms, all sorts of different things coming out of that same sort of approach. But ultimately, we're trying to pick information out of these spectra. If you were here for Dr. Brian LaPointe's talk, this is another example of the things you can get from remote sensing, from ocean color remote sensing. When you have that global coverage, we're able to see the Great Sargasso Belt in the ocean and how it moves and how it really took off in around 2011 and has stuck around since then. So this is, I think Brian showed a very similar movie. But basically, it's just the concentration of sargassum. And because we have so many, we have these sensors that are measuring constantly, we can actually get very high resolution looks at these processes. So this is, for example, how the sargassum is actually moving seasonally. So the first image was looking at it at sort of the peak in July of each year for a longer term history. This is the more fine scale movement of that sargassum in the ocean, and just really amazing views that we can get from ocean color, as an example. Okay, this is a little more recent. This is end of January into February of this year. If you were down here, you might recall it was, there's New England terms for how cold it was, but they're not for mixed company. But basically, we had that Arctic system move through, really pretty severe winds, very cold weather. And this is just an example from one of our ocean color satellites of what happened after one of those storms. And basically, with that sediment resuspension, you can see from before the storms, we have sort of typical look of the tip of Florida. And then after those storms, you have a huge amount of material that was resuspended off the Florida Bay there. And that sediment is actually very similar to those coccolithophores. It's carbonaceous sediments that are fine and very reflective. There's a lot of physical stuff that was going on that moved them offshore, but ultimately it's sediment resuspension. And then on we're using another sensor coming off of the Keys here. There's just amazing structure that we can see from ocean color and all the hydrodynamics, the physics that's going on. So it's really an incredible amount of information we can get from these ocean color sensors. And this is a great example of the backscattering of light. Here's another really interesting example, keeping it around Florida. This actually goes back to Hurricane Irma in 2017. Very powerful, incredibly destructive storm. The panel on the left shows Irma as it's coming in. Here's Florida. So it comes in and it's incredibly destructive. And on the left, you can see, other than destruction, some of the stuff that was left behind. This is showing the northern coast of Florida into Georgia. And coming back to that idea of tea and organics, there was such a huge amount of water flushing out of the land, through rivers and stuff. We were able to see these very distinct plumes of incredibly dissolved organic rich water coming out of these river systems. And there's major impacts related to carbon cycling and just all sorts of different things why you would want to know about that material heading out into the ocean. But just really stunning visual, and because we again have so much Earth observation out there, we're able to see all those sorts of things. Okay, keeping with hurricanes, this is just a segue into sort of, this is Hurricane Erin from last year, and this is actually from an instrument called NASA's PACE instrument, or Ocean Color instrument on the NASA's PACE satellite. Actually, several people here, researchers here at Harbor Branch, worked in both developing sort of the approach for that satellite mission and are still working on algorithm designs. Dr. Twardowski here, and I think I saw Alberto, Dr. Tonizzo. So I show this basically to give you the sense that we can see these storms over really wide areas of space, and we get pretty frequent revisits at this scale, so that once this storm is moved through, we can then go and actually look at spectra, like to see potentially how this storm might have affected the ocean in other ways. So, and this is just an animation of what that PACE satellite looks like. It's we call a sun-synchronous polar orbit, so it's always staying sort of in line with the Earth and the sun. And we call these things, it's basically making these stripes we call swaths. And it revisits any given location basically at the same time every day. And it takes 2 days to get global coverage, so it's pretty amazing. But that global coverage kind of comes with a couple of drawbacks. One is that it's also incredibly spectrally. Like these spectral maps, we get the full spectrum from 400 to 700 nanometers and beyond. So we have tons of information. It gives us a possibility that we might be able to understand phytoplankton and things like that. But the pixel size, so just like thinking about a digital camera, the pixel is 1 kilometer in size. So that's too big to see in a lot of different areas, like for example, the Indian River Lagoon. So just to throw this up here and give you a sense that these are some of the ocean color sensors that we use routinely. And there's a trade-off between the resolution, for example, PACE being a fairly large pixel, like 1 kilometer, and some of these other instruments like Sentinel-2 and Landsat that have much smaller pixels. So a 10-meter pixel, 30-meter pixel, that's something we can actually use in the Indian River Lagoon. We can look at dynamics and things like that. However, because the pixel is so much smaller, I don't know what happened there, we can't cover the same area. So, for example, the revisit time is how often a satellite will see a given location. And so for something like PACE or these other global scale missions, they're designed to basically revisit the same spot once every day or two. These much finer resolution, spatial resolution satellites only see the same spot every, let's say for the Sentinel, 10 days, or Landsat 16 days. One of the approaches to sort of get around that, though, is that, for example, there are 3 identical Sentinel-2 satellites that are up there and 2 identical Landsat satellites that are up there. And even then, you can also combine, their resolutions are similar enough that you can come up with a harmonized product that sort of combines all 5 of those sensors together. And now all of a sudden you've got you've got 5 very similar instruments with 10 to 16-day revisit times, and now we can actually get revisit times of satellites with incredible spatial resolution every, I think it's about every 2 to 3 days, 1.5 to 3 days. So it's pretty incredible, but it's still once a day. That's the problem. To give you an indication, a little bit of a tour for some intuition about the spatial resolution, why it matters. This is just a hypothetical 330-meter sensor, and you know, you can kind of see that maybe it's a coastline. I've looked at it enough that I know it's a coastline, but you know, as you increase the spatial resolution or make it finer, smaller pixels, you can start to see these features, and now you can actually start to see maybe there's an inlet, some different sediment types, water types. And then you get down to the scale of like a 30-meter instrument like Landsat, and now you can see very clearly. And so you can think about that in terms of, to put it in our own backyard, in the Indian River Lagoon, this is a Sentinel-2 image at 10-meter resolution of the area up around Cape Canaveral, Port Canaveral. And this yellow box here would be a PACE or other 1 kilometer size pixel. And so we would lose all the information. That would be 1 pixel. Zooming into that 1 pixel, the smaller grid here is 300 meters. So that's the Sentinel-3. It's a 300-meter pixel. You know, at 300 meters, we still get closer to the coast. We don't lose quite as much information, but it's still so much less resolution than in the 10 and 20, 30 meter resolution instruments. So we really need that spatial resolution in a place like the Indian River Lagoon. The revisit time is also a problem. So a lot of the phenomena that we're interested in happen quicker than every day, right? We're interested in things like runoff and storms that happen over hours, not weeks or days. One solution to that is you have a satellite that's in a fixed position looking down. And this is an experiment, or not experimental, it was a satellite launched by South Korea that has a geostationary look at ocean color. And you can see a lot of different features popping up. This is 1 day, and it would do 8 hourly snapshots per day, spaced an hour apart. And you can just start to see currents move and things like that. And that is the kind of features, the kind of resolution that we would ideally be able to see in the Indian River Lagoon, systems like the Indian River Lagoon. This is a figure that I think is really cool because it's a few of the ocean color sensors that have been up there since 1978. This is sort of the birth of ocean color. And then there's a whole bunch of different sensors since then. These examples here, they're all really big. They're expensive. These are space agencies sort of putting all their eggs in one basket, spending hundreds of millions of dollars to put a sensor together in a mission, and you kind of cross your fingers and you do all the engineering you can to make it work for your best shot. But you know, rockets still have issues. There's still mechanical, electrical problems and such. But again, really expensive, really big. But then on the left, you see next to the little stick guy, there's this sensor called Seahawk Hawkeye, and this was actually a CubeSat, which is a really small sort of satellite platform. And this is basically very similar radiometrically to this guy next to it, CWIFs. But this was developed for all the development, and the cost of everything was basically $1.7 million compared to our flagship global ocean color sensor right now, which is PACE. That whole development was around $805 million. And that doesn't get us into the Indian River Lagoon. This guy, the Seahawk, is actually really cool, Seahawk Hawkeye. These are some examples from some of the images that it's captured. It's not as high a resolution as we would like in a place like the Indian River Lagoon, but it's 120 meters per pixel, roughly a 3-day visit, few spectral bands. But one of the keys is at that cost, you can make dozens of these. Like we all watch the Starlink rockets go off, and they just, send out dozens of these satellites, these small satellites at a time, we can do something similar with ocean color sensors. And then we get that coverage. We can have enough up there that we're getting views of someplace like the Indian River Lagoon or coastal Florida where we care about those quick dynamics. We're seeing, multiple times a day. And I really like this quote from a NASA engineer that was involved in the launch and looking at the data afterwards. He said, "It's not bad for a shoebox," which, it's a very small little 4-kilogram thing. But one of the things I've become really excited about, this is inspired by some stuff that Dr. Mike Twardowski has talked about a lot. They worked on developing similar small platforms or small sensors similar to the CubeSats, but hyperspectral. And he mentioned the idea of putting these on these high altitude pseudo satellites. So they're basically a low power, high altitude drone. And they can stay aloft. They can linger in an area for weeks to maybe even months. And they could just circle. We could have these circling over like an area of the Indian River Lagoon, constantly looking at the ocean below. And I think we really need to look at solutions like this, where we either have ocean color sensors on balloons or on these sorts of platforms where we can get coverage of that's both high in spatial resolution but also temporal resolution, so that we can see blooms developing, so that we can see runoff from storms happening in more real time. So there's a lot of advantages to this sort of approach, and I think we just need, I don't know, $5 or $10 million to make it happen. We'll figure that out. So there's a QR code at the end. This is just a little bit of a palette cleanser of another ocean color image because I think it's really cool. There's these little white blips aren't clouds, they're actually sea ice. And the sea ice melting is really nutrient rich, so it's feeding phytoplankton as these little icebergs are melting away. I just think that's just beautiful. You can see the different colors, the turquoises into the greens, just amazing. Okay. So I'm not going to dig too hard into this, but this cartoon here has a lot of arrows and a lot of stuff. But basically, it's a way of looking at a process we call the biological carbon pump, which is the idea that microscopic plants, these phytoplankton, are in the surface. They're photosynthesizing. They're developing biomass, and when they die, or get eaten or clump up, they move that carbon biomass that they've made, then sinks out of the surface ocean. And there's a million things that happen to it after it sinks out of the surface ocean. This process is hugely important. It's one of the various arrows and lines and stuff in this diagram are some of the key questions in oceanography, biogeochemical oceanography right now. But our satellite sensors only see the surface, the euphotic zone, and even then they get an integrated view over depth. So there's a lot of useful information we can glean from that related to this problem, but ultimately we really need other platforms that let us look at properties versus depth. And not talking about it today, but a lot of the work we do is developing sensors that fit on these other autonomous platforms. And they still use optical properties and such to do, and maybe that'll be the part 2 lecture. Looking at that. I wanted to just finish with just one last marble. I think this is also really cool. This is the black marble. And it's Earth at night with one of these, I think it's VIIRS that measures this as well. But it's just really cool. We always see ocean color happening during the day. We're thinking photosynthesis and light things, but when it's dark, the currents are still moving stuff around. There's still all those phytoplankton hanging out, respiring, and doing all sorts of different biological things. All the different other critters and sea creatures are doing things. Zooplankton are migrating without the sunlight. But I just think this is a really interesting view, and I think we can still, there are technologies that are out there that can still get us information at night when we don't have the sun. We can use, active lidars potentially in the future. There are these floats and gliders that are still able to make measurements with active sensors at night. But another sort of takeaway here on this is, you also see the lights from human presence here, and I think this is a really beautiful way of seeing how close we are to the ocean, just as humans in general all across the globe. Most of this light is next to a big dark ocean, so I think that's really cool. Yeah, that's it.
Great job, Wayne. Thank you very much. You had some really cool videos and imagery. It's neat to look at the world from that perspective.
Razzle-dazzle.
Yeah, you razzled and dazzled us. Absolutely. All right, everyone. So we have time for a couple of questions. Do we have any? Okay, in back there. I'm going to paraphrase them for those listening online.
Because I always forget.
Yes, go ahead.
Yeah.
Let me repeat the
go ahead.
question for those online. The question spurred from the Hurricane Irma point, and the question was, is there data missing after major disturbances like storms?
So Irma. Yeah. So, I wanted to try to sort of sell ocean color here. One of the key limitations of ocean color is clouds, right? You can't see the ocean underneath this cloud. And once the hurricane passes, you'll often have clear days, but you still always have limitations with clouds. So certainly, if there's more storms, I think this is a classic Atlantic hurricane photo where there's Irma, and then I think Irma is the second of 3 that hit, one right after another across the Atlantic. Actually, yeah, because here's one right there. So yeah, clouds are bad, but in a hurricane like this, that's important. But almost a bigger thing is we want to use tools like ocean color to understand dynamics like the North Atlantic, for example. There's almost always clouds over the North Atlantic, almost the entire year. So you have very limited views of ocean color in those sorts of situations. And that's where these other platforms where we put out floats that have sensors that are always going all the time, they can measure ocean color potentially, but there's also other sensors that they could be deploying to measure different properties that we're interested in.
All right, next question. Ooh, does the technology
I didn't want to include anything about that. Yeah. There's thinking that yes, it could, in the same sort of sense that if the concentration's high enough, you might be able to see a signature, and maybe not with really large, a 1 kilometer global pixel looking at the open ocean, but maybe there's, certainly with a very high resolution imager, you could see, for example, rafting debris and things in particular areas. But there's some colleagues that are looking at how light interacts with microplastics in the ocean, and whether or not it might have specific either spectral or polarimetric signatures that are reflective of plastics. Yeah.
All right, one more question. Does anyone else have one? Or you get a double today. All right, go for
twofer.
it. How does AI help with data analysis and rapid response?
Yeah, so I like to joke that I was doing neural networks for this sort of thing before they were cool. But the core problem has always been the same. A neural network or a lot of these AI tools are really only as good as the data that's used to train them. And the way that training is done and the types of data that's included in sort of your data set, your training data set, is incredibly important and very difficult to do correctly. It's easy to, like, let's say we build an AI algorithm to try to understand, different types of phytoplankton in the ocean based on remote sensing signatures from something like PACE. If that training data set is just data that we've lumped together, it covers some range of conditions. Things like climate change or, different things move the system in a different direction, that can make our model that we developed using AI not as applicable to a new set of situations. So it takes a lot of care to do that stuff right, and there are groups that do, but there's also a fair amount of sort of slop in people attempting to do it.
All right, everyone. Do you have another question? Okay, our last one. Go ahead. The topic is still AI.
Follow-up AI.
Follow-up AI.
Is AI fallible? Yes. Yeah. I won't go too much into a lot of different personal opinions about large language models and things like that. But it's not an immediate solution, in my opinion. We can't just take all of the data and everything we have and throw it into a model and expect to get good results. You need capable training and understanding to put stuff together. You can't just put it into the wood chipper.
All right. Our very, very last question.
With the hurricane, the eye gets very big. If one of the sensor satellites were right there, would you get images through?
Can you get an image through a hurricane if you pass over the eye?
Yeah, I bet. Yeah, absolutely. I mean, I can't think of an example off the top of my head, but if the eye was big enough and the spatial resolution of the satellite was narrow enough, I think so.
All right, everyone, before we all disperse, Dr. Slade will be in the gallery with the cookies, and he can answer any additional questions. Please make sure you all come to see us next week for our graduate student lecture. And with that, join me in thanking him for this wonderful presentation. See you all next week.